In our final session today, we'll take a deeper look at some of the exciting ways that artificial intelligence is evolving in healthcare delivery. For example, we'll learn about how connected imaging instruments might enable new methods of image acquisition and image reconstruction in the cloud, and how AI models might speed up image analysis and reporting. We'll also explore how biomedical natural language processing is making it possible to extract population-level real-world evidence for precision health. Let's get started.
Hi, my name is Michael Hansen, and I'm with Health Futures at Microsoft Research. Today I'd like to talk to you a little bit about work that we've been doing on connecting medical imaging instruments to cloud computing resources. The reason why this is interesting is that we try to imagine what would happen if imaging systems were connected to unbounded computational resources. What could we actually do? How could we change the medical imaging experience? This is something that we can consider because medical imaging instruments are in fact not cameras. They usually acquire raw data in some kind of raw data space. In this case, we're looking at MRI here, and so data is acquired in what we call K-space, and it goes through a process that we call image reconstruction to then form the image. So if there are data missing from the data set as the data is being acquired, or if data is corrupted in some way, for instance from patient motion, then we end up with an image that has artifacts in it and is not diagnostic. But what's interesting here is that we can actually recover either missing data or corrupted data if we have lots and lots of compute available. So effectively there's a trade-off between difficult data acquisition and the compute that we have available, so that if we have lots of compute, we can recover a diagnostic image in the end. So looking at this at a slightly higher level, we could think about the things that we can deliver to a patient in a sort of simplistic way, being limited by what the instrument can do, of course, like how fast are the imaging gradients, if it's MRI, what's the field strength, so on. What is the patient willing to tolerate? How long can they lie still? Can they hold their breath, and so on? But it is also very much limited by what we're able to calculate. So if we have unbounded computational capabilities, we can change the experience for the patient. We can create a shorter scan, so less time in the scanner, or we could create a situation where maybe the patients wouldn't have to hold the breath, which they otherwise would have to do. So, of course, the question is, how do we do this? And we don't deploy the compute directly with the instrument locally. What we try to do instead is that we try to connect the imaging instrument to lots and lots of cloud computational resources, where we're then able to process the signal, do signal processing, do image reconstruction. And this may sound like a little bit of a crazy concept because of course there's lots and lots of raw data coming out of these instruments, but it is in fact not science fiction at all. This is something that we've been able to do for quite some time. Here's a very simple example of us actually scanning in real time, live streaming all the raw signal, the K-space signal, to Azure, doing the image reconstruction there, and then bringing the images back to the scanner live in real time. And that is done in a way that's completely transparent to both the patient and the user. Of course, there's lots and lots of engineering to be done to make this resilient, robust, compliant, and also to pick the right applications. So that's one of the things that we're busy working on. And one of the example projects that we've had has been a collaboration with Case Western Reserve University and Siemens, where we have tried to put a technique called MRI fingerprinting, 3D MRI fingerprinting to be specific, and do the image reconstruction in the cloud. So I won't go into details with this technique here, but it's something that allows us to map the tissue properties in various different parts of the body in a way that's very fast and accurate, and that allows us to improve diagnostics. But the reconstruction is very difficult, so it's hard to deploy this technique. So together in this partnership, we have then come up with a way to actually be able to do this in practice. So what we have are components that we get from Siemens that allow us to stream the signal out of the MRI scanner in real time. In the cloud, in Azure, we then have an open source image reconstruction framework called the Gadgeton, where this reconstruction is implemented in. And then we have a bunch of orchestration that we have put together in there that allows us to scale all of this up when we need to and provide the correct compute at the right time. And that means that this technique can now be deployed in many different clinical locations and tested out. Now, what we've also learned from this project is that it doesn't always go that well. So for instance, many of these prototype systems that are out there are brittle, they can fail, and when they do, it often impacts the experience that the patient has. So there's a lot of engineering here that needs to be done to get to a system that is much more robust. So what we're working on is resilient buffering that allows the scanner to come and go if connections break, more sophisticated orchestration of the compute management of all the artifacts, models, storage, and things that we need in the cloud, and also the whole developer experience. How does one develop new methods and actually bring them into this sort of image reconstruction cloud? So we will have much more information about this in the future, but that is part of what my team is engaged in. And then, of course, when we get the data to the cloud, we then have to think about what kind of cool things can we do with it. There's, of course, lots and lots of traditional signal processing, motion correction techniques, and other things that we work on. But we're also very interested in machine learning, and specifically using machine learning for doing image reconstruction. This is a topic that's been picked up in many labs and companies around the world. And just to sort of outline what this actually is, we would normally in MRI, if this is an MRI example, but in MRI, if you acquire a fully sampled data set, your image reconstruction would be a simple Fourier transform. Then if you sort of undersample the data space in some semi-random way, you get an image that looks not diagnostic. It has lots of artifacts in it, it has blurring in it, and so on. But if we instead use a model that we have trained on a large number of fully sampled data sets, we can actually recover an image with diagnostic quality that can be used. So there's been a number of different teams and activities around this, and challenges and so on, that have really tried to drive this forward. But what we've been looking into in more detail is, what are some of the places where this doesn't actually work as well as we might have hoped? For some of this work, we have built on work that's been done by Facebook and NYU in what's called the Fast MRI Challenge. And they've put out a large amount of raw data that the public can access. And this allows you to work on different image reconstructions. You can see what the images would look like if they were reconstructed from fully sampled data, but also with various other reconstruction techniques, including model-based reconstructions and machine learning-based reconstruction. But what was also learned in these early challenges was that when we do this accelerated MRI reconstruction using machine learning models, while we get images that at global level look quite nice, they're trained with what's called structural similarity index, and so they look as a loss function, they look really nice. But if we go into detail, we can see that there are actually some things missing. So for instance, here in this case, we have a meniscal tear in the knee. And when we look at some of the reconstruction, some of the top performance in these reconstruction challenges, these things actually go missing. And of course, this is highly problematic when we think about this as a diagnostic tool. So in order to be able to allow us to investigate that a little bit further, but also to allow the community to work more on these problems, we actually took this fast MRI data set, and then we had a bunch of radiologists, a couple of radiologists label the images so that we know where there are lesions. So we took about 1,000 knee data sets and about 1,000 brain data sets for which we have fully sampled raw data, and then we have put bounding boxes on all the different lesions that are in these images. And this now allows us to zoom in and kind of look at what happens in those regions where we might have something of clinical importance. We've taken all these labels actually and put them out on GitHub. So if you're interested in working on this problem, these are available out there. You can just download them. And actually, the folks at Facebook that maintain the FastMRI source code repo, where there are example models and so on, they've even created loaders to allow these annotations to be loaded. This now allows us to train models that can detect lesions. So here's a simple example of training a YOLO model to detect meniscal tears. And we can see that we now are starting to move towards something. This is not perhaps a very useful diagnostic tool, because we don't have great guarantees about actually picking up these tears, in this case meniscal tears. But what is cool about it is that it allows us to work very systematically and over a large amount of data to see what happens with different reconstruction methods in terms of our ability to actually detect these kinds of lesions. So we put together a full end-to-end pipeline, also based on this Gadgetron framework that I mentioned before, where there's a module that does a deep learning-based image reconstruction and forms the image. And then we also have a deep learning-based lesion detection module here that allows us to pick out these tears. So now end-to-end, we can reconstruct images and actually pull out this diagnostic information. And this now allows us to evaluate lots of different reconstruction methods here. So as an example, here we're looking at a rate 4 accelerator. It's a pretty moderate acceleration. We're acquiring a quarter of the data. We can see that a model-based, a machine learning-based reconstruction is able to quite nicely reconstruct the images here and show us the lesions that are there, and our automatic detection is able to pick up these lesions. But if we go to higher acceleration factor, which is actually where we probably want to be for this to really have high impact, we can now see that these lesions go away and our automatic detection of them also fails. So if we look at this across lots and lots of these data sets, we can actually see that, for instance, at 8-fold acceleration, we only detect about half of the lesions we used to detect. And so the ability to actually pull out small details of very high diagnostic importance really falls off here. But now we also have something that can point us in the right direction of where do we maybe need to look to train these models a little bit better. And specifically, you could wonder, what happens to this structural similarity index that we use to train these models? Is it different when we're able to detect and when we're not able to detect? So here we're comparing the true positives and the false negatives that we have in terms of the structural similarity index of these images. And we can actually see that they're pretty much the same. So the reason why these lesions go missing is probably because this is not a very sensitive thing for preserving those things that we would like to preserve in the reconstruction. But it allows us and others to start looking more in the right direction of where we can get these models to be better. So just to sum all of this up, we believe that we can improve the patient experience by connecting imaging instruments to more compute. On my team here at Microsoft Research, we're developing the technology to allow those connections to be reliable. Using that platform, we're able to bring scalable compute and machine learning models to the patient experience, but we still have a lot of work to do to improve this framework and also machine learning based image reconstructions. And with that, I'd just like to say thank you for your attention.
Hello, my name is Ozan. I'm a machine learning researcher at Microsoft Health Features. Today I would like to talk about our recent work on multimodal learning for healthcare AI. And in particular, I'll be talking about how we learn to relate radiology reports to medical imaging data. My talk will be structured around 3 topics. First of all, I want to talk about why we're actually interested in multimodal learning and how they can find use cases in clinical workflow optimization. And later, I want to talk about the technical challenges associated with building machine learning models in this space and how to be mindful of the unique challenges and how we can actually build pre-training strategies to address them. And lastly, I would like to show you a demo of our model trained on historical clinical data without requiring expert labels to solve clinical predictive tasks and at a scalable level. So to begin with, I want to show how the radiology workflow is structured today. So when we look at the radiology scans, quite often radiologists are expected to look at the frontal and the lateral views of this chest scans, as you see at the right hand side, and identify the findings and the change over time. And the findings are later summarized in the impression section, and recommendations are given to the referring physician. However, when we look at the current situation, we see that there's a growing backlog of medical image reporting, and this puts pressure on radiologists and increases the patient waiting time. To address this challenge, researchers have been recently looking into building machine learning models to assess this clinical workflow and optimize it through either second reading or patient triaging. But when we look at these systems, we see that they quite often require a vast number of manual annotations, and they're quite difficult to obtain because of time demands, and also experts' time is quite often very precious. And even if we acquire labels, we see that the existing ML solutions are often limited to a fixed set of abnormalities or body part. So how do we solve this challenge? What we see is that our domain is basically limited by the availability of publicly available data sets or large number of annotations. And quite often we don't have access to pre-trained models for object localization or text understanding. And when we look at the radiology sentences, we also see that they're quite often more challenging than the general domain captions, as you see in this slide. For example, a simple bag of word approach wouldn't be sufficient to identify the key labels in terms of the severity or the progression or the laterality of the findings. So to address this, basically one needs to be mindful of the text modeling aspects and how we can actually get the most out of this data. In particular, when we look at the data, we see that there are negations and long-range dependencies between the entities. Quite often we wouldn't see a caption saying there's no dog in this picture, but negative labels can be actually useful to build robust and accurate machine learning systems. And also we need to be mindful of text unrelated to medical scans. For example, it wouldn't be an optimal solution to try to bring these representations with images because they're actually not shared across the two modalities. So we need to find ways to filter out this information. To address some of these challenges, in our recent work, we looked into how we can get most out of the text data, which is due to be presented in October at ECCV conference. And to start with, what we looked into is basically, can we build domain-specific corpora, ranging from clinical notes to PubMed abstracts, and we build a domain-specific bioclinical vocabulary. And this is trained with this domain-specific data using the standard procedures as you see in the phase 2. And this canonical model is publicly available, and the canonical model is in the third stage, is fine-tuned to obtain better representations for the chest radiology data. So for this, we basically deploy text-only self-supervised learning, where we try to match the information between the findings and the summary section to bootstrap our system further and also obtain better sentence representations, which are later to be used in joint learning to supervise the image models. So for this, how do we actually achieve this joint training? So we first pre-train the text and the image encoders, as I mentioned earlier, and now we perform joint contrastive learning between these two modalities at the final stage. And this joint contrastive learning basically operates on the global representations of images and text data, and the framework can also be used in conjunction with local contrastive losses, such as patch to token embedding matching. And the representations obtained at the end of this training can later be used for various applications, including unimodal evaluation and joint multimodal evaluation. However, we need to be mindful of the benefits of and the what we actually answer when we do these evaluations. And quite often when we look at the existing work, we see that researchers have been mostly focusing on unimodal evaluation, but we believe that this does not answer the question whether we're actually making the most out of the text data. And this is quite important, especially when we have limited annotations and limited data. So to address this challenge in our work, we basically propose a new benchmark that is related to phrase grounding, and we name it as MSCXR, which looks at various aspects as you see in this table, including image text encoding and how well they align in this joint latent space, and whether they can actually localize the correspondences between tokens and the patches. So I would like to show you how this benchmark looks like. So for this, we build a demo, and this is publicly available as well. Here we will basically go through some of the examples collected from the Mimic CXR dataset, and we will try to ground some of the clinical findings in the images. So these clinical findings will be passed to the model as text queries, as you see in here. For example, we are testing this model with cardiac sulate is enlarged with this prompt, and we should expect some response between the patches around the heart region and this text query, which will be shown at the bottom right here with the red color, as you see, and the isocontours in the middle. This system can be also tested with various other queries, because it's trained with historical data that should be expected to have holistic understanding of different clinical findings, such as consolidation, chest tubes, and also other findings. So in this example, we will be basically testing the system with the query of consolidation, and we will explicitly emphasize upper lung lobes, and we will also add a modifier that is bilateral. So we should expect response on both sides of the lung lobes and the upper region. And these annotations are basically all curated by 2 expert radiologists, and we released 1,162 instances of them in this benchmark. And in this final example, what I would like to show you is that when we type in left pleural effusion and right pleural effusion, we should expect the model to understand this modifier information, the lateral texture that matters. And when we type in left, we should expect the model to respond on the left lung lobe, which is shown on the right-hand side of the image. And similarly, when we type in the right, that should focus on the basal and the costophronic angle on the right side of the, left side of the image, as you see here. To give further details about this benchmark dataset, I want to emphasize that we have collected these annotations by working closely with 2 expert radiologists, and they're all verified. And these sentences are basically collected from the report, so they're not artificially created, and they have quite complexity in terms of the graph structure. And we also covered a wide range of findings in this dataset. When we look at the quantitative results, we structured our experiments in basically 2 groups. In the first group, we looked at how well do we do this in the phase grounding tasks. And we looked at 2 particular metrics, contrast to noise ratio and intersection over union. So the more density is basically accumulated within this bounding box, the better the performance is expected to be. And what we see is that we use CXR-BERT and compare it against other text models using the exact same setup in terms of imaging and also in terms of hyperparameters. And CXR-BERT basically consistently leads a better performance when we look at this phase grounding results. And it can also play a complementary role to recent advancements in the field, such as local contrasted loss based training. And the two can be combined, and it can lead to even further improvement, as you see at the bottom row. And in the second set of experiments, we looked at unimodal evaluation of this foundation model. And here we were trying to answer the question whether joint training actually helps these individual text and image models in their downstream tasks by means of transfer learning. And in the first set of experiments, we looked at how well does this text model perform in terms of sentence embedding and sentence semantics, understanding of them, after this joint training. And what we see is that the NLI, natural language inference performance, actually improves quite substantially when we do joint training and also do pre-training with these models. Similarly, when we look at the results on the imaging side, we tested the downstream classifier on pneumonia detection dataset, and we see that the demand for the manual label training data is significantly reduced, as you see with this blue curve, and also it leads to better predictive performance. Our work is publicly available if you're interested in playing with the demo which we showed you earlier, and also the source code. Please visit our GitHub page, and also the models are publicly made available through Hugging Face. And if you'd like to learn more about the benchmark dataset and the associated paper, please visit the links at the left-hand side. Lastly, I would like to thank my team for the great work and efforts they have put in this study. And throughout our research, we've been working closely with clinical experts, which help us to focus on the clinical impact. And our team has been basically composed of natural language experts, imaging, and machine learning experts. So through this multidisciplinary effort, we are able to build this foundation model. If you have any questions about our work and any suggestions, please feel free to reach out to us via email. And if you would like to learn more about our current ongoing projects, please check the Teams page highlighted at the bottom. Thank you.
Hello, everyone. I'm Hoifeng Poon from Microsoft Health Futures. Today, I'm very excited to be here along with Dr. Carlo Bifoco from Providence. Together, we are going to update you a little bit about our recent progress in advancing AI for precision health. The advent of big data, such as genomic sequencing, as well as electronic medical record, herald a precision health era where we hope to substantially improve clinical care efficacy as well as clinical research efficiency. However, big data also present the grand challenge of information overload. For example, the explosive growth of knowledge and complexity in precision treatment actually may further exacerbate the clinical care burden and inequality. Clinical research is already very expensive and slow, and the problem may worsen now that we seek to develop more treatment to precisely target ever smaller subpopulations. So at Microsoft, we aim to advance biomedical AI to help democratize care and accelerate research. In the past decades or so, we have been focusing on advancing foundational research in biomedical NLP powered by self-supervised learning. These advances help us to start make some initial progress and incubate end-to-end applications with key stakeholders from clinical providers to pharmaceutical researchers. A key focus in our research agenda is machine reading. Given a piece of text, we want to extract the key biomedical entities such as gene, mutation, drug. And more importantly, we want to extract key relations such as that a tumor with certain gene mutation may respond to a certain treatment. We have made a lot of progress in developing accurate machine reader to extract PubMed scale knowledge graph from biomedical literature. And more recently, we increasingly focus on extracting high-definition patient information from electronic medical record for scaling the generation of population-level real-world evidence, or RWE. Having this longitudinal patient journey from diagnosis to treatment to outcome, RWE may actually be able to substantially help improve care and accelerate research with many immediate low-hanging fruit in the clinical trial and post-market space. For example, Flatiron has shown that by curating RWE from electronic medical record, we can simulate a virtual control arm to help speed up label expansion. And for this reason, it was acquired by Roche for $2 billion. However, like many other RWE vendors, Flatiron rely on hiring an army of medical specialists for curation. The US alone have 2 million new cancer patients every year, and curation can take hours per patient. So in order to scale to population level RWE, we sorely need AI and NLP technology to help empower this kind of curator to become superhuman and drastically accelerate curation speed. The standard supervised learning paradigm for NLP unfortunately requires a domain expert to annotate a lot of input-output example, which become the major bottleneck. Instead, we have been pursuing the alternative paradigm of self-supervised learning, where essentially we're trying to leverage free lunch resources to bootstrap learning. Specifically, we have developed many effective methods to leverage available domain knowledge such as ontology, existing knowledge bases as blueprint to essentially automatically create lots of noisy examples from unlabeled text. For example, we have explored domain-specific pre-training and showed that it can be highly effective for biomedical applications. Our work on PubMed has become the de facto standard in biomedical pre-training, outperforming even language models that are much larger, pre-training on much bigger general domain text. We have proposed a deep probabilistic logic, which can serve as a unifying framework to combine noisy and potentially contradictory knowledge sources for self-supervision. And most recently, we have explored incorporating contrasted learning as a powerful way to support knowledge-rich self-supervision. By using just the UMLS ontology and the unlabeled PubMed text, we were able to train the CRISPR model, which is essentially a universal entity linker for all 4 million UML concepts, outperforming prior state-of-the-art by up to 20 points in accuracy. So building on top of all these advances, we start to make some inroad in end-to-end applications as well. Without any label example, we were able to develop accurate machine reader for extracting precision oncology knowledge, and preliminary studies show that this can already help speed up knowledge extraction, knowledge curation for a commercial product, the JAX CKB. On the RWE side, in collaboration with Dr. Carlo Bifoco from Providence, we were able to develop a NLP pipeline for structuring all incoming cancer nodes and start to power end-to-end applications such as molecular tumor board and clinical trial matching. Building on top of domain-specific pre-training and deep probabilistic logic, we were able to quickly develop new NLP capability. For example, in tallying real-world evidence for immunotherapy against tumor mutation burden. Previously, this would have required thousands of hours of manual curation as well as manual annotation of tens of thousands of supervised examples. Going forward, we start to expand beyond NLP and start pushing on new research frontiers such as multimodal learning, causal inference, privacy-preserving AI, which are all essential for advancing RWE and precision health applications. I want to acknowledge my amazing team who actually do all this work, as well as our key collaborator, both within Microsoft and externally. Next, I'm very excited to have Dr. Carlo Bifoco from Providence, who will demonstrate some of the end-to-end application from our collaboration.
Yeah, thank you, Hoifung, for the kind of introduction. I'm very excited to be here today to be able to demonstrate the practical application of the NLP work and machine learning work that we have done together. What I'm showing you today is in clinical production, and I think is pretty much the state of the art of the field in terms of clinical production right now. So again, super excited to be able to demonstrate that. If you look at my screen currently, you will see features of a patient. This is coming from our tumor board interface. Tumor boards are the standard of care for treatment oncology. You can see that we can display information coming from the EMR in live widgets, which can enlarge and diminish, where we have biomarkers and other relevant information coming from pathology and radiology. And so forth. And particularly, you can see that the pathology images are fully dynamic and available for consumption in a live environment. The same is true for the genomics information. What I'm going to do now, I'm going to deeper a little bit in the genomic findings themselves and show you how the power of NLP and machine learning really enables us to very quickly capture essential pieces of information for the clinical treatment of a patient. So what we're seeing here is the results of what we call comprehensive genomic profiling. It includes multiple biomarkers which are relevant for the treatment of a patient. The patient we are currently considering is a patient who has a diagnosis of lung adenocarcinoma involving the left upper lobe of the lung. He has, as you can see here, multiple genomic alterations. The most important one is highlighted here. It's an EML4 ALK fusion, and that is a very exciting finding from a genomic perspective, because it represents a biomarker that is actually actionable and can be targeted with precision medicine. In particular, this particular biomarker can be targeted with what we call a Class 1 or FDA-approved drug. So it's an exciting finding. It can really change the life of the patient and have a dramatic impact for people who are otherwise often incurable. So if I can give you a little bit more information about this particular biomarker by looking in the clinical trial interface that we have built with Hoifung and the team. You can see here the biomarker, and you can see here multiple trials which actually match that biomarker. In this case, the target is really the ALK fusion. And information about that is available in this interface, again, based on the work that Hoifung and the team at Microsoft that developed, really enhancing keywords and enabling me to identify very quickly key information that I will need to include or not include the trial in my curated annotation of the patient. So an exciting finding, and everything looks very, very good here. But let's go back to the genomic findings and examine them a little bit more carefully. And you will see that in the genomic findings, there are multiple alterations in the gene called ALK. There's one here, and there's another one here. And that finding is a little bit perplexing. It's not usually something that we identify early on in a patient's course. And so what is going on here? How can we sort this out a little bit better? Well, one pathway is to take advantage of the work that Hoifung has done on the clinical trials, but I showed you before, but also on the annotation of the EMR wider body of data. And let me show you how that really works. In this tab, I'm now zooming into information coming from that environment. And you can see that we're dealing here with a patient who is relatively young. A lot of information here is hidden because of the need of annotating, of hiding annotation, which is PHI, but you can see that we're dealing with a young male, as I mentioned before, an adenocarcinoma of the lung. If you scroll down the findings, you will see that actually we are dealing with a lesion that is unfortunately recurrent. And let me give you some more information about that by scrolling the annotation that we're getting from the EMR. You can see here that there are multiple automatic classification schemas for stage, I'm going to go now on what marks for M. And what a machine learning algorithm is really doing here is highlighting information which maps the staging in the electronic medical record and highlighting all the terms which are of significance. And when you see me scrolling this, you will see that the patient had previous treatment, also received therapies for the ALK clone, and particularly in this case, the lower blood TNIP drug, but then actually failed to respond. And so what we're dealing here is with CNS mets, which are progressing, unfortunately. And you can see how I can gain all this information very rapidly just by taking advantage of this interface. So this is telling me, A, that a patient was a responder, did benefit from the therapy, and it's also explaining me now why we are dealing with this mutation that I showed you before, this particular one, which is actually associated with resistance. And that can explain the progression of the brain met. So now that we have this piece of information about the resistance mutation, what can we do? Well, we have the opportunity to take advantage again of the clinical trial matching engine. And that's what you're seeing here. You can see the mutation I was describing before in a clinical trial setting. And so what this clinical trial is actually showing you is the ability to target the emergence of resistance through this pathway. And here's the further deep dive into that one of the particular clinical trials which can help the patient I was demoing you before. So the interface is incredibly powerful. It enables me to gain at a glance a critical piece of information that will drive the treatment of the patient, even in a complex environment like the one I showed you before. So this is very exciting. It's in clinical production, as I mentioned before. It makes a difference in the treatment of our patients every day. But beyond that, it really enables us to move to the next stage, to the future. And the future is really multimodal machine learning that Hoifung has alluded before in his slides. And so what you're seeing here is a foundation of data extraction from the EMR and from genomic data sets. But that, I think, is just the first step of a multimodal fusion, which will incorporate machine learning done on images coming from radiology, from pathology, and from different environments, which we feel is going to be really the future driver of personalized medicine. Why I'm saying that? Well, if you think about this particular case that I showed you now, the responses are temporary. And they're often not fully understood. And so what we will have in the future is the ability to actually incorporate additional layers of information, like tumor microenvironment, et cetera, which are really going to be driven by the images I was mentioning before. So very exciting future, really foundational work. And again, thank you for the opportunity of demoing this to you today.
Hello, and thank you for joining us in the Health and Life Sciences track of the Microsoft Research Summit. What a fascinating and exciting time we've shared together. At Microsoft Research, we are optimistic about how technology can shape and deepen our understanding of fundamental biology and drive the development of new tools for clinical medicine. Today, we've seen how our community of researchers, scientists, engineers, strategists, and medical professionals are working with our partners at the frontiers of science to bring the highest levels of knowledge and skill to the twin challenges of reimagining health in the life sciences and empowering every person on the planet to live a healthier future. The research and projects included in this track are just a small part of the work Microsoft is doing, and we invite you to stay connected to us and to our work through the Health Futures Forum, where we continue to explore technologies and challenges in health and the life sciences. We also invite you to join us for the other tracks of the 2022 Microsoft Research Summit to learn more about our work in other areas. Thank you for joining us today, and we'll see you around.
That's a wrap for the 2022 Microsoft Research Summit. These few days have been an opportunity to step back and discuss the impact we hope our work will have on the world. It's exciting to come together like this for the kinds of conversations that happen whenever thoughtful people gather and engage. We hope that these discussions and collaborations will carry on throughout the year. See you next time.